Yeah, go ahead. Sorry.
All right. So I'm going to talk about mathematical computational modeling in biology. And I really like this cartoon. It's about mathematician trying to explain the results, and probably a pure mathematician. You see, the question is, tell us in layman's terms, like easy way, what your breakthrough means, result. And the answer is certainly, of course. And a mathematician gives a formula, right? And it doesn't explain anything to people. It might be simplified formula. It's a special language, right? So there is a language barrier between mathematicians and scientists and engineers. And some people understand some of it. Most biologists, they don't understand mathematical language. So I'll try to explain what might be done from my point of view, from my experience, to reach out to biologists and make simulations relevant to biology. So I'll be talking about general subject, which is called mathematical development or mathematical theory of development. Just want to mention a workshop which we organized with Christophe Gadin, Philippe Mainy, and Roland Merckx at Leiden University Lawrence Center 4 years ago now, well, maybe 3.5, specifically kind of to revisit different opened questions and problems in this field. And there are quite a few conferences on this topic being held every year. Okay, so what is biology? How is biology different, say, from physics? Biology studies living matter. So it deals with cells. Some types of behavior of cells is governed by physics, chemistry, you name it. But it's a living cell. So it grows, it divides, it actually changes shape, it can move, it can start building extracellular matrix. Cells can aggregate, okay? They can start dying, so going through apoptosis. And everything happens at specific moment in time when you look at the developing embryo. So development means study of studying development means studying embryos. And embryo starts with a couple of cells, right? And then they start dividing, and these are all stem cells, cells which can differentiate into any cell in our body. And then cells aggregate, then genetics kicks in. Some of them start differentiating, they become differentiated cells. They form organs, and then a baby forms, right? Embryo forms, and after a certain period of time, the embryo is completely formed. It's kind of proportional. Every organ is there, and it starts growing as a whole. And then the baby is born, and so on. And this is pretty robust. Of course, there are so-called birth defects, right? Genetic defects, which result in babies being born with disfigurement. Sometimes babies are born with 6 fingers. This is indication of severe birth defect, which manifests itself in other ways. So this is why it's really important to study the embryo development. At the same time, a lot of effort being actually spent now in biomedical engineering. People trying to grow organs outside of embryo, outside of human body, and then maybe implant it back. People are talking about growing human hearts, implanted in animals and putting them back in humans. In our group, we're actually trying to grow pieces of epithelium to put back into the heart after the heart attack. I'm just trying to justify importance of studying development, right? Also, when you look at the embryo, kind of follow the way life evolved on Earth. So initially, there were just individual cells kind of crawling around, bacteria or microorganisms. And then they started aggregating, aggregates formed, and then small animals. And here we go. Now there are we have humans with the brain activity and the rest of it. So if you look at what happens inside of an embryo, cells do very specific kind of things. They go through specific processes. As I said, apoptosis, proliferation, division, cell fusion, locomotion, cell can move, so stem cells can move. Chemotaxis, hypnotaxis type of motion. Cells can move towards maximum like chemical gradient. Cells can adhere to each other, and then they can form small pieces of tissue, and tissue can fold. And this folding stage of development of tissue in an embryo, that's what I'm going to talk about here. All right, so there are kind of huge hurdles, obstacles to studying developmental processes mathematically and computationally. Not everything is known about cells. Then there is a huge amount of noise in what's, and this is what your group has understood studying, and what individual cells do and how signaling is performed. Still, the system is kind of robust. On the average, a lot of cells do the same thing. When you implement a mathematical model, and I would call it mechanistic model which tries to account for mechanisms. On a computer, it's very expensive, actually. The more detail you put in, the higher is the computational cost, and I'll talk about it a little bit. Comparison with experiment, that's been a really kind of weak point for a long time. Now with development of machine learning approaches and AI, that becomes actually doable if enough experimental data is available. The outputs are huge if we succeed in understanding development and kind of modeling it. So it might create a very important tool for synthetic morphology, creating tissue outside of human body and putting it back. And actually just kind of manipulating biological mechanisms to our advantage. And also development processes are not that different from what happens with cancerous cells. All right, so let me talk about several multiscale models. I'll explain what multiscale means in this context. So this so-called cell-based models, models which represent individual cells at very high resolution, actually are called cell-based, and they can be classified in lattice-based and off-lattice-based. So lattice-based, it's when your cell moves on a fixed lattice, okay? And off-lattice, you don't have that kind of assumption, and I'll talk about a model like that later. So let me talk about cell-Lopot's model, which is pretty popular in biological community. It's a generalization of Ising model from statistical mechanics, all right? But Ising model, it's a model for magnetic material. You have two types of spins, spin up, spin down, and clusters are forming, phase transitions are studied, the rest of it. The main focus of that model is to find global minima of your system. So when we want to model things in biology, models are not supposed to be at equilibrium. So what I'm going to talk is going to be generalization of Ising model, but it's non-equilibrium. So each cells are represented by several nodes on the lattice, and we can have different types of cells, 1, 2, 3, they're different. And then we can have maybe a couple of cells of the same type. So I can have like a cluster of pixels, like type 1 being close to this one and so on. And then extracellular matrix or substrate is represented as this substrate underneath all these clusters moving in it. Boundary conditions can be introduced as periodic boundary conditions, so a big lattice can be chosen and so on. The important consideration for this model is that it's a Markov chain model, right? So state of the system, of this dynamical system, is kind of this distribution of nodes which are occupied, and then there is a multidimensional index attached to each node which is occupied. So you need to know that this is a cell of type so, like 1, and that's a cell number 3 of this type, for example, if there are several of them. Well, there's this one big cell called extracellular matrix. It's not a cell, it's a medium in where cells are actually propagating. And then we can calculate energy of actually adhesion energy of interaction between individual pixels on the boundary of two clusters. So it's called adhesion energy. And oops, somehow, so there is a Hamiltonian, energy Hamiltonian. And what happens with this model, we go from one state to another by choosing at random a node and flipping the multidimensional index. So for example, at random, say I end up at this particular location. And can you see my cursor?
I'm not, no, we can't. Can you move a little bit? No, unfortunately, we cannot see. Maybe.
I annotated it.
Yeah, maybe you can click the left bottom of the PowerPoint and you can change it to the laser point. Then maybe we can see more clearly.
Unfortunately, I cannot. I don't know how to do that. But anyway,
Yeah.
I'll try just to explain it then.
Oh, Michelle said she can see the drawing. Yeah, they said
Okay.
annotation is okay. Yeah, it looks. Can you try one more? Oh, yeah, I can see cursor. Okay, yeah.
Okay.
Yeah, cool.
All right. So you choose a specific location at random, and then actually you flip it. And then you recalculate the energy. This one flip changes the energy by so much, but that's the result. If the energy decreases, so the change of energy is like negative, you accept it as probability 1, because you want to get to the local minimum. But if it's still positive, you accept it with this probability. It's called Gibbs measure. Why? Because you want to travel to flip a lot of pixels and get to local minima, but then in the end, you actually want to get to the global minima. So if you hit a local minima and there is no kind of non-zero probability of getting out of that local minima, then you're stuck. This is why once in a while you're allowed with certain probability to get outside. And you use generator of random numbers to actually to take this into account. So this is detailed description of Hamiltonian. So there's a contact energy, just basically if you have two pixels representing parts of two different classes, two different cells, there's the energy of interaction, which is kind of given. And then you recalculate it when you change the type of a pixel. So if you do it many, many times, there's a danger of actually ending up with a cluster kind of going to 0 for particular cell representation. So to avoid that, you introduce so-called target volume and target area for each cluster. And then what you get in the end, your cells, suppose you want to model kind of Brownian motion, just move at random and the volume or area fluctuates a little bit, but never goes to 0 and never goes to actually to be too big, because you have this constraint. Okay, you can introduce a different lattice and couple it with a chemical field and introduce this into Hamiltonian, and you'll get a chemotactic behavior. So this was applied early on to model early stages of development of a chicken limb. The chickens have three digits in the end. But what's more important, the arm, whatever, the limb starts with 1 bone, splits into 2, splits into 3. And we were able to use this model, and it's multiscale, because chemotaxis is involved in production of chemoattractant by cells and fibrinogen and so on. And we got kind of dynamical growth of this pattern. And you're actually looking at individual cells as well as distribution of chemical there. And it goes through bifurcations once in a while.
Mm-hmm.
And we kind of found a perturbation which resulted in abnormal number of digits, as you can see, 4. And the same perturbation, actually, it's a perturbation of a biochemical system, signaling system, is actually might be linked to what happens with humans as well. So this is a structure multiscale model, different levels of description, so molecular level, diffusion of extracellular, intracellular chemicals, and that's what one of your collaborators mentioned, right? Subcellular level, it's a cytoskeleton dynamics, microtubules, actin, actomyosin dynamics, genetic regulation, cell differentiation. Cell level, it's what happens with cells. They divide, they differentiate, they adhere, grow, they can die, interact with extracellular matrix. And you put everything together and you get cell actually living with other neighboring cells in a specific environment. So in case of limb development, cells actually grow, they move a little bit, and then they use chemotaxis to move in specific direction, they adhere to each other, and then at a certain point they differentiate for genetic reasons.
Mm-hmm.
So this was all done on a lattice, and modeling on a lattice might introduce certain artifacts into the model. But there is a way to deal with it. Also, to criticize this model a little bit, it's not that easy to represent mechanical properties of evolving tissue, because these are cartoons. They don't account for specific mechanical properties of cells, but they are kind of easy to implement. It's Monte Carlo, and not easy to parallelize, but they're pretty popular, and there are packages available for that. So what does multiscale mean? So different biological processes actually are operating at different scales, as we just discussed. And everything gets unified in order to produce certain function, say function of development in an embryo. But from a mathematical point of view, multiscale means connecting partial differential equations to numerical schemes, to kind of going down to discretization. And that's a classical kind of subject in applied mathematics. It's called numerical methods, right? You have a PDE, which you usually derive from first principles in physics, okay? And then you discretize it. You try to solve these equations using numerical schemes. You discretize it. So, and then you can go further and discretize it even more. But there is a different way to establish connection between, say, discretized systems and PDEs. And this is what people do in physics. They start with systems of interacting particles, Brownian motion, right? And they derive in continuous limit, specific time in diffusion limit, they derive diffusion equation, Laplace equation mathematically, right? And what we did for cellular pods model, we tried to derive continuous limit for that system. The difference is people like Kelly and Siegel, they derived their classical equation, reaction diffusion equation, from system of interacting particles like in the early 70s. It just, their cells were represented by particles, one cell, one particle, like agent-based representation. Here, cell is represented by extended object, a cluster of pixels, right? It has characteristic size. And the question is whether this can be taken into account when deriving continuous limit. So you're looking at derivation in one-dimensional case. There is one-dimensional cell consisting of like, I don't know, 1, 2, 3, 4, 5 pixels. And it's kind of going through random walk in one dimension. That's the Hamiltonian.
Mm-hmm.
And then we need to have a restriction constraint on the length and of a cell, and this interaction with extracellular matrix on the right and the left. And this is so-called actually master equation. So we're going to come up with mean approximation for this Markovian dynamical system, Markovian process. So we need to introduce so-called transitional probabilities of going from one state to another. So what is the state? The state is a cell being kind of positioned over a certain number of nodes, specific nodes, on a one-dimensional lattice, well, semi-axis here, on a substrate, okay? And how do we calculate it? So the main variable here is the probability of finding a cell of length L at the center of mass at X and at the kind of specific moment in time. So let's introduce like time t and next time point, and the time step is epsilon squared times delta t, because we need to go to continuous limit. Okay, I'm trying to derive continuous limit for this system. Let's see what kind of equation we're going to get. Okay, so suppose that the cell was a little bit longer on this side. There was an additional pixel here, and then it turned into substrate, so it lost one. So we have a probability of having a cell, let's see, L plus, well, one of these probabilities, just probability of having a cell of length a little bit, well, this one, length was bigger. You see L plus, and then the step of the lattice, again, we're going to introduce epsilon delta X there. We need to find the right scale for that, because epsilon will be sent to 0 in the end. And for diffusion limit, you need to kind of have epsilon squared kind of order and epsilon for time and space, so on. And then we introduce transitional probability, but that's the probability I just demonstrated for the Potts model. So you kind of flip it, there is delta E, change of energy, and there is acceptance probability for that. Either it's 1 or it's exponential to delta E, and that's exactly this probability. So we have this master equation written here, and then we're going to look at continuous limit when the ratio of this two kind of time step divided by X step squared is going to be kind of fixed.
So sorry, Mark, could you tell me what capital L exactly mean in the picture?
Large L? Large L?
Yeah.
Large L, it's a constraint for the length of a cell.
Oh.
So it's average length. So if it overshoots it, there will be like probability of this kind of extending even more is going to be reduced because of the Gibbs measure. If it gets smaller, the probability of actually adding additional pixel will be increased. So
Okay,
each step of the kind of dynamical system, we cannot predict what's going to happen. On the average, after like many millions of steps, we will get a pretty clear description. All right, so and that's what we get. We get this equation for the probability distribution function for density, which can be solved using this ansatz. And it's just a probability of two functions. One represents distribution of lengths, and the other one distribution of positions of centers of mass of these cells. And it's possible to substitute and separate equations. And actually, distribution of lengths is nothing else, just the Boltzmann distribution. And kind of time distribution or dynamics of actually the density of centers of the cells actually satisfies Keller-Segal. And of course, in the process, we will have in here, actually, we're going to count for L. L is going to show up in the distribution, in Boltzmann distribution. So it's parameterized by average size of a cell, say in your experiment. Okay, we did it in two-dimensional case as well, for rectangular cells. And then Keller-Segal actually equation has a problem. When you couple, say, just diffusion of these particles undergoing kind of that kind of random walk with, say, chemotactic potential, which looks like this. So there is a maximum, it goes like this. It's going to result in all particles going to the same place, to the maximum. And killer seagull actually has a blow up in finite time. It gets an infinite and finite time, which is definitely not biological. Why? Because we shouldn't be modeling cells as particles. They have volume. So we implemented so-called exclusion volume principle in derivation of the equations, and we obtained so-called nonlinear diffusion equations, which are parameterized by characteristic sizes of cells, and it's 1 over p squared somehow. So basically, you get a nonlinear diffusion coefficient. So when cells get too close to each other, on average, the size of individual cell, they stop, they slow down. And that's what this nonlinear diffusion actually does. And we did it in 3D. And this is, of course, chemotaxis equation, which is coupled to reaction, well, diffusion equation. And this is diffusion, but it's nonlinear diffusion. And then a lot of people in PDEs started looking at these equations. Now they've been generalized, actually characterized, studied. And this is just kind of a set of simulations relating celopods model simulations to continuous limit. And under certain we can actually calculate certain types of patterns, steady states for this nonlinear diffusion equations, reaction nonlinear diffusion equations. And the parameters chosen for getting these patterns can be projected back on celopods model. You get very similar pattern as well. So multiscale means going from continuous description to discretized, starting with discretized description going to continuous, or some processes happening inside of cell or piece of tissue can be represented by using discretized representation and some partial differential equations. These are so-called hybrid systems, multiscale hybrid systems. So I'll give you an example. Let's see how much time I have.
You still have about 30 minutes?
How much?
30? Half hour?
Okay.
Yeah, please take your time. Yeah, don't worry.
I don't want to rush. So I talked about mathematics and multiscale modeling and what it has to do with biological classical modeling. Now I'm going to try to talk about biological relevance. Okay, so development of a limb is an important process, but it's kind of happens in animals. There is more classical kind of biological system, which been studied a lot. It's a development of a wing of a fruit fly, so-called Drosophila wing, imaginal disc. And that's what we've been focusing on with our collaborators from University of Notre Dame for more than 15 years now. So you all know what the wing is, right? This is how it looks like. But it doesn't look like anything like this in the initial stages of embryo development. So embryo initially, and I don't know how much you know about that. It's like, actually, that's the embryo. That's the picture here on the left. And there are these discs on the surface of the embryo, which are going to give rise to development of specific organs. So the front area of the embryo is going to kind of fold inside, and this is how the head is going to develop. And the one in the back, if I'm pointing to the right sides of the embryo, are going to fold in, and actually that's beginning of the actually track inside of an embryo, and so on. But wings kind of extend away from the embryo, from these discs here. Whoops. Okay. And look, if you look at the fully developed wing, it's very delicate, right? It has this so-called veins, has this cartilage, and size is very important, and shape is very important. For example, if there are two wings develop and one is bigger than another, the fly is going to die immediately, right? So it won't be able kind of have a good control of the flight and so on if they don't grow to be too big. So the question is how it's starting with like just a very well-packed piece of tissue with a large number of cells. This develops into this structure in 3D, which flattens in the end. And I'll try to explain how this happens and kind of what kind of mechanisms are involved. And so we'll discuss that in a second. So, so the whole development takes hours, and initially there are like 50,000 cells. It goes from 50 to 50,000 cells. And it's pretty robust.
I see.
Okay.
Mhm.
They develop like millions of them, like non-stop. And in the lab of our collaborator, they've grown non-stop. And the lifespan is not very long, but still you can kind of image their development and actually take all the images. Now people are trying to measure the mechanical stresses and so on. Genetically, it's been known, the genome has been known for 20 years. It's one of the most studied biological systems in the whole field of biology and development. People use fruit flies for studying human cancerous cells, genes. They put them in flies and look how they operate, basically. It's very kind of predictable system, let's put it this way, because signaling has been studied extensively. But when you start putting together signaling with genetics, with mechanical properties of cells, control of growth becomes complicated. Okay, so let's try to look at, so let me go back and just explain, look at this part of the embryo, this imaginal disc from a top. So I'm going to talk about modeling apical view and side view. And let me start with the apical view. This is how it looks. So it's not in real time, but you see cells actually kind of adjust. Some of them become kind of rounded, become very large, and then they kind of get squeezed again and so on. So what happens? So cells actually, they're so-called columnar cells, they're very elongated in this part of an embryo. So they're elongated and their nucleus, it's kind of constantly moving inside of each cell. And when there is time to divide, nucleus goes up to the boundary. Can you see a cursor or not?
I can't.
And some people kind of
Can other people can see the cursor?
You can see cursor, right? So I'm
oh,
looking...
now I can see it. Yeah.
Okay. Oh, why?
Yeah,
we can communicate, right?
I think it disappears, and if you move it, then it appears, I think.
Oh, that's right. That's right.
Yes.
That's right. So I'll be moving it.
Yeah.
So,
okay.
so you see this apical view, that's what this movie is all about on the left.
Mhm.
But underneath, you don't see it. These cells are very elongated. And nuclei go up when cell needs to divide. Because if it sits in the middle somewhere, there's too much pressure coming from neighboring cells. It cannot divide. It needs to grow, divide. So it's much easier to go up. And how does it go up? We don't know. We just, from apical view, we just see once in a while, because of the nucleus going up, that the cell expands. It's called rounding. And then you see it divides into two nuclei appear, they go down, and they get squeezed, and that's it. So if you look at it from a mathematical point of view, it's a kind of, it's a puzzle. Cells with different shapes feeding together. And it's been studied heavily, apical view of a wing during development. So people used finite element models, vertex models. Vertex models are kind of simplified representations of cells from the start. Each cell is represented by a certain number of nodes and edges. It's like Voronoi diagram. And then they just, there is a way to introduce mechanical stresses nowadays, but it's not straightforward. And then we use so-called subcellular element model, coarse-grained molecular dynamics. representation of individual cells. I'll talk about this in a second. So cells are represented by different types of nodes, quite a few of them, connected by springs or interacting with each other using specific potentials. So it can Morse potentials. So they get attracted at characteristic distances. When they get too close, they get repulsed and so on. And then there are nodes representing different subcellular compartments. Like we have nodes representing actual liquid inside of a cell, or representing internal pressure in the cell, or nucleus for this matter. And this diagram basically describes the setup. So each node has an equation describing a so-called Langevin equation. And usually there is a kind of random noise term added here, like Wiener process, just to account for like random fluctuations. So when cells are present in the tissue, they don't move too much. They don't do this a lot, okay? They just kind of sitting there. So we kind of neglected the stochastic term here. And x dot is just derivative, kind of velocity of motion of each node in this complicated representation. Interestingly enough, it's possible to calibrate this model. So the parameters are basically spring constants, and there are some other constants present in the equation here in each type of energy term. Because there are characteristic kind of distances, I told you, these are parameters of the system, it can be turned on. So people have been studying in physics, biophysics, studied mechanical properties of cells for a long time. They take two cells, individual cells, using optical tweezers, kind of see them adhere, push them towards each other, they adhere, and they start pulling on them. And then they can measure the force it takes to dissociate them. And we mimicked that kind of measurement in our simulations, and we were able to get very close to what was observed by actually choosing specific values of parameters. And the model has a minimal number of parameters because, you know, the deal, right? The more parameters you have, the easier it is to fit with any data. So that's like was one of our first simulations of this rounding on apical view. These big cells, they kind of grow and divide. And you see, this leads to a so-called polygon distribution. This is how it looks. And mathematicians got really excited about it because they can classify them. There are polygon classes. There are specific laws which were kind of introduced for kind of analytically predicting the number of cells of specific type, say. So here the kind of can be hexagonal or pentagonal and the rest of it. So statistics is very important. So we were able actually to run our simulations and reproduce what is observed in experiment. So I showed the experimental movie, right? So segmentation can be applied and shapes of individual cells can be determined and then calculated, and statistics can be calculated and the rest of it. All right, but that was like more kind of, how to put it, physics-driven result. Biologists are not that interested in shapes of cells. What they're interested in, it's like what happens with a piece of tissue in general. How these shapes or divisions taking place in specific part of the tissue at specific time all come together in determining a specific shape of the tissue. And of course, everything's controlled by signaling. And of course, people have been looking, different groups, into coupling mechanical representation for cells. And I just talked about mechanics. I didn't talk about signaling yet. And distribution of chemicals. So there are packages which have been developed over the years. So one comes from Alber's lab. So it's this one. And then there is MegaGene and Virtual Leaf and so on. So we recently actually published a paper on a new way to couple mechanical submodel, subcellular submodel with a chemical one. So basically, we introduce triangulation based on shapes of individual cells, and these shapes, they're not prescribed. It's not like a vertex model, right? We get what we get. If we take our representation of a cell out of the tissue, it's going to become spherical. It goes to equilibrium, and the equilibrium is spherical shape. So the shapes, the morphology, the shapes of cells, that depends on interactions between them, on adhesivity properties, mechanical properties of cells, whether or not they're dividing. And then, of course, there is a layer of extracellular matrix underneath, and they're all attached to that. So it leads to distribution of stresses and so on. So here with this model, which couples reaction diffusion equation, partial differential equation, right, with a system of interacting particles, and this is what subcell element model is, okay? And they're coupled in space and they're coupled in time. So diffusion is very fast, right? And so solving PD is very fast. What happens with cells after they start responding to certain kind of concentration of chemical in part of tissue takes time. So in response to like increase in specific signaling, and this is kind of description of the signaling submodel here with different types of chemicals present during development. So it takes them time, and they can start dividing more frequently as a result of high levels of certain chemical present in specific part of tissue. And what we were able to show, and that project was led by Dr. Wei Tao Chen, a colleague of mine from UCR. So we were able to show that actually the gradients of morphogen, some of these chemicals, actually control the growth of the tissue, the final size of the tissue. So in this case, there are no boundary conditions. It's like kind of experiment envisioned outside of an embryo, and that can be done basically. And we were just testing kind of coupling of chemical submodel with mechanistic submodel. And we were comparing with certain experimental data as well. That's a simulation. You see kind of this rounding, cells dividing, right? But you also see distribution of the chemical. And the way this chemical kind of spreads and introduced controls the shape and size of this piece of tissue. And that comes pretty clear, close to what people see in experiment. All right, so we were looking at, yeah. Okay. So we looked at the apical view. Now let's look at the side view. So apical, you just look from the top, and then suppose you can cut it. And this piece of tissue is pretty kind of, there's radial symmetry there. So you can cut it in different ways, and it's going to look more or less the same. Okay, now you see the cells, right? I was telling you that the cells are very elongated, and this is how they are, columnar cells. And here, when you look from a side, of course, this is confocal microscopy. You have scans. These are three-dimensional images, which we can look at, basically. And you can tag different parts of a cell. So myosin, nuclei is fluorescing in blue, integrin, and actin is present here. So actually, by determining where the green is, the actin is, you can see the membranes of individual cells. And then you can see myosin, and you see there's a concentration on top, and sometimes there's concentration on the bottom. And then integrin, it's responsible for adhesion. Mm-hmm. So if you look at the side view of this piece of tissue of the embryo developing wing in the middle of it, you realize that there are two layers of ECM on the bottom, basal, and on top. And this is actually the diagram of this piece of tissue. Columnar cells, and then there are this squamous cells on top. There's a layer. So here the cells are elongated. Here they kind of all kind of squished and cover the piece of tissue. And what happens, cells in response to the distribution of chemicals, which produced by individual cells in response to other cells, basically start bending the whole piece of tissue. And you can actually determine the distribution of concentrations of some of the chemicals there. And at least you see what happens with cytoskeleton, and the whole thing bends. And the curvature of the surface is evolving curvature is extremely important. If it doesn't get to specific curvature, it's not going to start bending in opposite direction, and the phase of the development is not going to be completed. So getting the right curvature for the piece of tissue during this phase of development is very important. You perturb it, you're going to get a fly with absolutely undeveloped wing. And of course, we've got like hundreds, if not thousands, of images. Our collaborators in the image analysis group actually characterize them. And you see a kind of distribution of different types of components, as I said, integrin, myosin, and actin in space, just based on the images. And shouldn't be trusting what I have to say. These are experimental images, confocal microscopy image. It's three-dimensional. You see individual cells, they're elongated, and you can actually guess where the nuclei is, and so on. So during division, these are the steps of division. That's what happens in individual cells. So, and that's what we were testing. So it wasn't clear. People kind of were thinking, just judging from the images, it wasn't clear whether this mechanism is the one which is important in division and formation of a curvature of a piece of tissue. So we kind of assumed, we were testing hypothesis that actomyosin present on the bottom part of a cell actually shrinks, contracts, and pushes nucleus up. It's like, like toothpaste. Remember? You kind of push it and it goes up. And that's the mechanism which pushes it up. Then close to the upper layer, apical layer, they divide. And from apical view, you see the surrounding of cells. This is what happens. We have a proof of that. And then actomyosin kind of now starts contracting on top and pushes them down again. And distribution, positioning of this nuclei, that's what determines in the end, basically, the curvature. Right. And don't forget that there's a layer of ECM.
사랑니 발치 관련해가지고 상담도 잘할 것 같으세요?
Excuse me?
Oh, I think someone is unmuted, so I just muted them. Yes.
Okay.
Yeah.
Is there a question? No?
I don't think so. Please go ahead.
Okay. All right. So the basic question was whether we're able to find some characteristic curvatures or development of coverage in time, more or less for wings on the average. And that's usually done using machine learning approach. And the answer is yes. Actually, yes, they all kind of go through specific changes in curvature during this stage of development. And there is some kind of fluctuation, but it's more or less the same. So there's a pattern of formation of the curvature, of the shape of this wing. And that's what we based our model on. We model motion of nuclei. We have actomyosin contractility. We have ECM. We have adhesivity of cells with ECM, adhesivity of cells with each other. And then ECM in our model doesn't play an important role. It plays active role, okay? ECM is something which is produced by cells. It's a biogel. It's a very important part of any tissue, but so we kind of assume that these are two basic kind of mechanisms: actomyosin contractility, motion of nuclei, and interaction with ECM. ECM is kind of spreads out the stress, because if individual cells starts dividing, nucleus goes up. it can easily leave the tissue if it's not younglet attached on the bottom to the actually ECM. Actually, there is a very thin layer of ECM on top, kind of holds the whole piece of tissue together. Stem cells in embryo, they are not very, very adhesive. Okay, they can leave the tissue once in a while. So what we concluded using our simulations, so basically this motion of nuclei, that's and their distribution in time, positioning, what determines the curvature. And ECM is very important because it kind of memorizes the shape. It puts it in place. So there are quite a few active cells which go up, divide, and so on. Curvature is forming, and it's bending, but it's because of ECM, it doesn't go back to the initial position. ECM, it's kind of absorbs the stress coming from individual cells and redistributes it for the whole piece of tissue, kind of brings them together. And then this is kind of, we suggested an experiment, okay? So we ran a simulation where we suggested to remove small piece of ECM and see what happens. And in simulation, it starts kind of, at the end, it starts folding back. And our collaborators ran an experiment. So they degraded layer of ECM, and they obtained shapes which are very close to what we observed. So it's an example of predictive simulation. And after that, the biologists started kind of really started paying attention to what we have to say. So mimicking what they already see in experiment is not enough. It's like if simulation produces new questions, new ideas for experiment, it's like biologists go, never thought about it. That's the best outcome you can hope for. Then they'll start kind of trying to confirm it in experiment, and they come up with new mechanisms as well. So that's the simulation, one of the simulations. And once again, there's a layer of ECM. There are these elongated cells. There are cells which kind of sitting on top. They're taken into account, but they don't do anything active here. And this is a removal of the ECM. You see it folds in opposite direction. Folds in opposite direction, and that was observed in the experiment. So this supports our kind of prediction of the role of ECM as kind of a youngster, something which holds everything together. All right, so let me talk about our recent result. And the paper on this subject was accepted like 2 days ago.
Oh, congratulations.
In Nature Communications.
Nice.
Took forever. It was a great effort on the part of students, 3 rounds of revisions, you can imagine.
Yeah.
We have 3 different mechanisms we are kind of testing in this experiment, both in simulations and in experiments. So here we take into account in detail the division of cells. There, I talked about motion of nuclei. We didn't quite take it all the way to division. So we looked at a very short period of time in development. Here, we really allow cells to divide, really made a model much more complex. And that's kind of data, again, we get. We can track individual divisions, and this is the level of resolution. It's pretty good. And so I'm not going to go into a lot of detail about it. I just want to show a couple of simulations actually set up and run by Jennifer, who is on this listening to this talk. So one simulation, it's kind of an in silico experiment. It's like what happens with this piece of tissue when we increase the ratio of apical and basal contractility. So we introduced divisions, and now we allow contractility of myosin on the bottom and on the top of each cell. And this is controlled in a specific way. And if this ratio changes, that's what happens. This is what happens with this piece of tissue. So there's this middle part, which actually doesn't change shape too much.
Mhm.
And that was, as I understand, was observed in experiment as well. And this is a tissue shape when you increase extracellular matrix stiffness.
Mhm.
Right? Measuring mechanical properties of ECM is doable, but only few labs can do it. So we're kind of predicting which specific stiffness probably is present in our experiments, the ones we're comparing with. And again, you see, the goal here was to get the flattening in the middle part of a wing, and it needs to flatten. I told you that it goes through sequence of profiles with specific curvature, and flattening is a very important stage in this kind of constant reshaping of a wing. This one, it's a simulation which takes into account increase in the adhesion of cells, these vertical cells, with the ECM. So if you increase it, and some of this kind of corresponds to us kind of looking for the right parameters.
Mhm.
And in some cases, we're trying to compare with profiles we see in experiments. And again, this comes pretty close to experiment. And the last one, it's tissue shape in response by changing pressure. So I told you there are nodes inside of cells, and they represent pressure, internal pressure of a cell, which is important kind of part of a kind of compartmental part of a cell.
Mhm.
The amount of liquid and the pressure there. You see, cells are dividing, by the way. And then again, we get the right type of shape and structure. A number of cells corresponds to what people what observed. Well, proportional to what people observed in the experiment. Our collaborators observed in the experiment. So in this paper, we tested 3 different mechanisms, and that's the result. And so the next step, we're coupling now this. We have another paper underway when we introduce coupling with a chemical signaling network. At this point, we just kind of looked at images depicting concentrations of chemicals in time without dynamics, and kind of allowed cells to respond that, and still got the right shapes. Now we're testing kind of coupling with a very fast PD model for like reaction diffusion of signaling, and that's the next step. And then the other project we're working on, we're going to look at modeling the surface, not in 2D, but like 2D surface in 3-dimensional space. And there, the mathematical challenge is solving Laplace equation on a deforming, evolving surface. And of course, if it's not flat, it's not a Laplace equation, it's Laplace Beltrami. So you need to take into account local curvature and actually Riemannian metric on the surface. And that's what we've been looking at. All right, so let me talk about members of the team. I was talking about the results. So Jennifer, this, and she's here. She was absolutely kind of a leader of this project on not just simulation or model development side, but also working very closely with biologists and bioengineers. Kevin Tsai, former student and postdoc, and he developed the first basic model for these processes. So Dr. Chen is my collaborator, and she's an expert in signaling as well as cell representations. Whoops. So Ali Nibaktash, former postdoc, developed a few first models, and also he was instrumental in developing a model for the apical view. Alicia Tomei is a former student. She just graduated. She's working on the signaling part of the model. Aliriza Mizani, coupled mechanical and signaling model for the apical view. And these are our experimental collaborators and friends. So Professor Jeremy Zartman from Notre Dame. And of course, Dr. Wei Tao Chen is a professor as well. All right, so Nilay Kumar, who is a doctor now, he graduated. He's a postdoc at Developmental Biology Institute at Purdue University now. Ran the experiments with Maisha Mim. And also, let me mention Maricruz Flores, who ran additional experiments for the paper. Well, thank you for your time.
Thank you. Thanks for a great talk. So everyone, it's a question Q&A time. So if you have any question, please feel free to unmute yourself and ask questions. So let me stop the recording.